Meta-Proteome-Analyzer.
Yeah, thank you for inviting me here. I come from Magdeburg, and I maybe talk about a use case for a graph database. It's bioinformatics, so biology and biochemistry, analytics. So a bit off topic here, but I will not go too much into the details of the biology here, but more focused on the actual implementation use case of the graph database. So life sciences and graph databases. Life sciences, as you can see, are interested here in genes. So the blueprint of life actually, expression of the proteins on the next level, and also on the metabolism, so the interaction of these proteins. Here you can see a protein interaction network. It's looking quite complicated, but this is what biology is about. It's not that easy actually. These life scientists and biologists are not interested in so much boring tables. So unfortunately, this is what you get your data. Mostly it's stored in Excel sheets, and as a bioinformatician, I always have to deal with these huge Excel sheets of several megabytes, or even if it's better, then it's an SQL database, but mostly it's Excel sheets. So the shortcomings are scalability and performance issues. You guys know this better than me. Complexity, the lack of information sometimes, and most importantly for me is that these models are far away from biology, from the biological reality. So just short to talk about what my topic is. I'm working in my PhD work on. It's metaproteomics, so it's about it's a protein research field which studies functional activities in microbial communities. So, for example, if you think of your human body, you have many normal human body cells, but you have 1 order of magnitude more microbial cells inside your gut, on your skin, and so on. So this is what this research field is about these microbiology inside different organisms, not only humans. So metaproteomics results, they can be used for taxonomic assignment to find out. So you have a sample and you want to find out what kind of species inside. You took seawater and you want to see what is inside this seawater, what kind of organisms. So the problem is always that you have one protein which is pretty much looking alike in the different organisms. So it's a one-to-many relation, and of course you have many proteins as well. It's getting even more complicated if you have short protein fragments. These are here on the left, the peptides, and one peptide may belong to 2 different proteins, and then this protein B, for example, can belong to 2 different species. So it's always one level on top of it. And then again, you even have more of these different entity types. So it's quite complex to handle, and we want to find a more natural model for this. So that's why we transfer this graph model of biological data back to the database storage level by using, here you have the biology graph, by using graph database. And that was the main rationale behind to decide for a graph database, because biology is already a graph. So we implemented this graph database system of Neo4j. We just took the free version, I think 1.9 we're currently using, into our software Meta-Proteome-Analyzer. And that is what the model is looking like, pretty simple for each of the entities. We have nodes. I don't want to bore you with all this, but we have also enzymes and pathways and topologies. And of course then you have the relationships which are interesting to query. So which peptide belongs to which protein, which species refers to a protein, and which of these proteins belongs to a pathway. So you get this kind of complex network already here, and that's what it's looking like for a new small sample. It's visualized in Gephi. We're not interested in the visualization, but just to give you an idea how complex this already can be. So you see different species in proteins and pathway here, lipid metabolism, and Escherichia coli, which is bacterium on top. But did you find that guy here in there? No? Me neither. But that's the important thing. Scientists claimed Tyrannosaurus rex peptide, which they would like to have found in a T-Rex bone by this method of shotgun proteomics. And it turned out, and actually many researchers then took this into question and found out that it's just a contaminant from modern species, because you have collagen and keratin, and it's a contaminant. For example, if you look in the lab and you put some hair inside your sample, then you have keratin. And it was obviously not the Tyrannosaurus rex, but it was published in Science. And these guys are still publishing the same stuff over and over again. What they could have done is they could have filtered out these T-Rex contaminants. That is what this first simple query could be. So here on the left side, you can see Meta-Proteome, that's a protein group, so it's just one sort of protein family which we just named Meta-Proteome, and you can filter out just by the name of the description of the contaminant. In this case is keratin, and you would filter out here what you can see all the keratin proteins of your data set. Of course, this is a very simple query, but it's quite handy to have it, and also quite flexible. And it's done by Cypher in our application. So the user can always change the question that he's putting into the data set and get different answers. So this flexibility, I think, is one strength. That's a more complicated one. We are also dealing with samples from biogas plants. So biogas is a regenerative energy source. And we want to find out what kind of proteins are working on the production of methane of biogas. So if we want to find out which of these proteins are involved, we can query just the pathway here. That's the pathway of the methanogenesis, so the production of biogas. And you can see then easily what kind of proteins and taxonomies are involved in this pathway. So that's the Cypher query. I think the guys who work on Neo4j know this, of course, and for the others, I think it's really easy to get. So you have a starting point, then you have two different match clauses for the pathways, and you can specify the return values in the order. In the application, I will skip this and come later. In our application, we're using this here for the queries, so you can just type in your Cypher queries in the console, but you can also do a composition of different match clauses, for example, here, and select the starting point just by drop-down menus. And also you can set the return values here on the right side. And you can save these queries and put it to your favorites. So the lab user who's not really experienced with informatics can easily access this kind of query language. So he doesn't, well, it's not as easy as he could just ask the question directly, of course, to an informatics guy, and he will find it out. But with this, I think composition, our lab users are trying to fit it into. So that's what it's looking like in the software, and just performance. I'm not a benchmarking guy at all, so I just took some really small data sets here, and what you can see if you query the same data set against a server, which we have as a machine in-house, then you can see that Neo4j is scaling quite nicely, I think, on the different data sets. Do I have still some time?
I still the tag is here in my.
Okay, so that's application what it's looking like, so you get some statistics and of course insight after asking the queries. Yeah, I think that's it. It's tested by different labs, and we are documenting and lining up the manuscript now. As scientists have too much time, you can see try the different logos. Yeah. So there was spinning.
Okay, so your test, you get to fully in time.
Thank you, Bill.
So, questions?
Yeah. So, on the SQL side, there's 40 years of history of trying to enable users to make queries. So, now on the RDF and graph side, there is obviously less history for doing the same thing. So, what's your experience in users actually modifying queries that exist, or let alone composing their own?
Yeah, well, we have 2 PhD students who are also working on this software, like as part-time job. So they're quite happy with it, but I'm not having really much experience with new people getting this hand on the tools.
Could you imagine an end user, like a researcher, customizing, somehow modifying a query, describing a relationship?
I hope, but yeah.
Tricky.
It's tricky, yeah. But maybe this kind of composition where you just drop down any entities, for example, could help there somehow.
I'd actually like to try that with some recent tooling we've built. Is the data available? Is the data available here as well?
Yeah. You mean the source code? Or? Yeah.
Yes.
Yeah.
In RDF or just in?
The data itself.
The data, yeah, with the data set on the website now.
But I'd have to convert it to RDF. Yes.
How large is it?
No, it's not in RDF.
Yeah, I have a few questions. So this is more my field. So just to summarize, you do mass spec, identify peptides, you get subset of proteins, and you want to do this for all of it, for all proteins basically, not all proteins, but be able to detect any random protein, assign a certain sub-taxonomy to it.
Yeah.
So basically, just to make sure that I'm getting it, so you start with nucleotides of mass spec experiment, for example, get some peptides out, try to make peptides. How do you deal with the fact that you have about 2 billion peptide combinations that you need to deal with at that scale just to be able to figure out which peptide you add in mass spec? Because that's a humongous explosion of entropy. Because currently you had experiment with 10,000 proteins. I hope you think I have 56 billion proteins. But if you explode the peptide search stage, you have about 2 terabytes of plain 6-character strings that you end up with.
So what we are actually doing is that we are storing everything on the central server. So all the experiments, so all your data will be on the central server, and we just query one experiment one by one. So we kind of reduce this thing for the graph database client. So we have all the data in there on the main server. We store it in just usual SQL, and then we just query the client with a graph database backend.
Okay, so basically saying that your peptide restricted model is outside of the graph database. So
Yeah.
first assumption. Okay, yeah. So basically it's a TUC technology.
Right.
That's quite key.
Yeah, because I think this would crash on terabytes.
Yes, I know. Yeah. Okay. The second one is, why did you go for, I saw that you use Unigraph data, or at least you showed tables of Unigraph data coming back. Why did you decide to use Neo4j and not take down download the Unigraph XML or RDF XML from
We actually query, after each search, we query all the different metadatabases, so Uniprot and Unigraph via web services. We're not using RDF.
I noticed that.
Yeah. So I think this would be another interesting step to go, but we haven't done that so far.
Okay, so currently you just do wget to uniprot.org.
Yeah. input API and cake pathways and so on.
How do you use the academic license and the issue?
So we have it as Apache 2 license now. This one.
No, cake, the database.
No, the cake we do not download the whole database, just query it after each run. Yeah.
And have you looked at other pathway databases like RIA or Reacta?
Meta-Proteome we have looked, but it's mainly for human proteins. So we had an implementation of the Meta-Proteome database once as a project, but as it was not usable for us, because it's just human proteins basically. It's a good database, but we look at human data.
But you don't work on human data?
Well, we work on human data in terms of human gut samples, but these are bacterial proteins. And that's what we're interested in, and this is not in this database.
So I'm a completely ignorant in the field of let's say, bioinformatics. Do you have, I think I've seen one of these cyber queries that represent one of these pattern matching that you do. Can you please go back to this?
Probably there's a mistake in it.
No. So I was wondering if this pattern matching, this pattern is searched for basically each vertex in your graph, more or less, right? So you apply this to each vertex, starting from each vertex and each reverse. So I was wondering, so this goes, I think, 1 hop or 2 hops away from the source, the furthest. It traverses like something like a close neighborhood. So I was wondering if it's common in this kind of workflows for to have pattern matching queries that go further, right? So even further, I would imagine that graph-specific engine is even more efficient for large scale workflow. You would need something that works at large scale even more. But if this is still simple, well, simple is a big word, but I was wondering.
So we come to match more clauses, for example, more entities, and also ontologies on top of it. So,
but do you have at any frequency something where you have a variable length path? Let's say you have a very deep hierarchy of something, and then anywhere within this tree or anywhere within so an unbounded number of steps in a graph. Does that happen in your workflows?
Visit a query.
It should, if you ask for something like or possibly gram-negative bacteria identified. Then you have quite a large hierarchy that you can traverse with like 500,000 nodes from that path expression.
yeah.
Right, because this is the kind of thing that we got to put in benchmarks. There's some work to be done on the database side for doing it efficiently. So we're interested in finding use cases which do have these features.
Okay.